So the first protein that we're going to look at today is ferritin. So ferritin is a protein that stores and releases iron. So it'll actually bind to iron ions and release them as needed. So it's actually produced by nearly all living organisms. So from humans down to bacteria, basically all living organisms express ferritin. And so what it does is it keeps iron in a safe, non-toxic form, and it acts as an iron buffer. So it can bind and release iron as needed. So that stops you from having an iron deficiency or an excess of iron. And so we're going to actually just look at the individual subunit of ferritin today. But so the individual subunits of ferritin are 20 kilodalton proteins that will actually combine to form a hollow sphere made up of 24 ferritin subunits. So right here is the individual subunit of ferritin, and right here is the 24 ferritin subunits that are combined to form this large hollow sphere that will bind and release iron. And so the structure we're going to look at today is the protein database code 1AEW. So it's L-chain horse apoferritin. So it's ferritin from a horse, and it's apoferritin because it's not actually bound to iron. So it was from Equus caballus, which is a horse, the organism. The expression system was E. coli. So this protein itself was expressed in E. coli, and it was deposited a long time ago in 1997 by this group here. And so it has a resolution. It was determined by X-ray diffraction, and it has a resolution of 1.95 angstroms, which is quite good resolution. So let's take a look at this structure. All right, so here we have the protein, a single subunit of the ferritin protein. And you can see here that it's made entirely of alpha helices and these loop regions here. You can see there's an extended loop region in the back of this protein here, but there are no beta sheets whatsoever. So ferritin is only made up of alpha helices. Now we can actually focus in on one of these alpha helices to sort of investigate it a bit more, and we can actually look at the side chains and the backbone, so the actual atoms that are making up this alpha helix. So in this structure here, of course, you just have the ribbon diagram, which shows you that there's alpha helices. That's a really good way to visualize a structure from the beginning. But those alpha helices are made up of individual atoms that are just forming that conformation. So we can actually see the backbone here. So the backbone carbons of the residues of the amino acids in this alpha helix here are now shown in yellow. And then here you can see the side chains, which are shown in pink or magenta. So for this view, the carbon atoms are colored yellow for the backbone, and the side chain carbons are colored pink or magenta for the side chains. However, the oxygen atoms are always going to be shown as red. The nitrogen atoms are always going to be shown as blue. And then if you choose, and then the sulfur atoms like in methionine or cysteine are going to be shown as yellow. So this actually gives us an opportunity to investigate firsthand what is holding those alpha helices together. And you can see from here that the as I talked about in the lecture, that the backbone of the residues in that alpha helix are actually forming that alpha helix. So you see that you have these connections between the backbone carbonyl oxygen and the backbone nitrogen in the amino group that are holding this entire alpha helix together from one side to the other. So that secondary structure, those alpha helices, are only being held together by hydrogen bonding from the backbone atoms. And you can see that the side chains are actually sticking out from that alpha helix backbone. So the backbone, the hydrogen bonding in the backbone are what's holding it together, and that allows the side chains to stick out from the side of the alpha helix. So while we're looking at the alpha helix, we can actually use this as an exercise to go through our amino acids so we can actually see which side chains are present on this alpha helix. So first, right here, we have a glutamate residue. And that's actually followed by a glycine residue. So you don't see a backbone in this region right here. And that's because it's a glycine. There's only a hydrogen in that backbone, and that's actually not shown in this structure. So that's followed by a valine, as you can see here. And then on the other side, we have a cysteine. And that's followed by a histidine. And that's followed by a phenylalanine and another phenylalanine right next to each other. Right here, we have an arginine residue. And then we have another glutamine. You can see the carbonyl group there and 2 methyl groups. And that's followed by a leucine. And this single methyl group sticking up is an alanine. So next we have another glutamate, followed by a second glutamate. And all the way in the back here, we have a lysine residue, and then we have an arginine residue. And that's followed by another glutamate. And then there's a glycine in the back, so you can't see that side chain. And that's followed by an alanine. After that alanine, we have another glutamate. So there's a lot of glutamate residues in this particular alpha helix. And that's followed by an arginine, as you can see here with this guanidino group. Next, we have another leucine residue, followed by a second leucine residue right in a row. And that's followed by a lysine. See, it has a long chain and an amino group at the end. In the back here, we have our first methionine. And that's followed by our first amide amino acid, which is a glutamine. And that's followed by an asparagine and another glutamine. And then lastly, at the end of this alpha helix, we have another arginine residue. So that's the ferritin, a subunit of the ferritin protein, and it's composed entirely of alpha helices with some loops as well. And we learned that we saw firsthand that the alpha helices are held together by backbone hydrogen bonding, and that allows the side chain to stick out from the sides of those alpha helices. So the next protein that we're going to look at is a fatty acid binding protein. And these are proteins that will bind and transfer lipid molecules from different compartments in the cell. So fatty acids or other molecules like retinoic acid or stuff like that. There's 11 known families of fatty acid binding proteins, and they are distinguished by which tissues or which organisms they operate in. So some will operate in muscle, some in liver, so on and so forth. But despite them being grouped into different families, all fatty acid binding proteins have a conserved structure. So this structure right here is going to be found in all the fatty acid binding proteins overall, despite whatever tissue or organism that they operate in. And so here is the actual deposit for the structure we're going to look at in a second. So its code is 1FTP, and it's a muscle fatty acid binding protein. And it was isolated directly from this desert locust called Schistosera gregaria. It was deposited in 1994 by this group, and it was determined by X-ray diffraction. And so that means that this protein was crystallized and then shot with X-rays to determine its structure. And its resolution is 2.2 angstroms, which is still quite very high. So you can see, compared to ferritin, the fatty acid binding protein is made up mostly of beta sheets. So there's one alpha helix that you can see here at the top. But other than that, it's made of this beta barrel type situation where it's sort of a cylinder of beta sheets, all interacting with each other to make the overall tertiary structure of the fatty acid binding protein. Now we can actually focus in on a couple of these beta sheets to take a look at what's going on there. So again, we can look at the actual residues or the backbone and side chain amino acids that are making up these two beta sheets right here. So all the carbons in this are shown in yellow, and then of course the oxygens are in red, and the nitrogens are in blue. If there's any sulfurs, those are in a darker yellow color. So we can see here that, as we discussed in the lecture, that the beta sheet is actually held together by hydrogen bondings between adjacent beta sheets. And of course, specifically, it's the backbone of the different beta sheets that are making these hydrogen bonds with the other beta sheet. So let's keep the backbone in yellow, and then we can change the side chain carbons to pink or magenta to see what's going on. Now, if we look over this side, you can see that those hydrogen bonds are between the backbone carbonyl groups, carbonyl oxygens, and the backbone amino groups of the adjacent chains, and that allows for that stability that makes the beta sheets. But also the side chains are going to stick up above and below those beta sheets, as you can see here from one angle and then the other angle. And this, of course, gives us another opportunity to practice our amino acids, so we can see the different amino acid side chains that are sticking above and below the beta sheet. So here we have a leucine residue, and it's going to be sticking down below the beta sheet. Then on top of the beta sheet, we have a valine. Below again, we have a histidine residue. And then at the top, we have a glutamate. And then at the end of this beta sheet, we have a glutamine residue. And then the one right next to it is a lysine. And so that's the fatty acid binding protein, or an example of a fatty acid binding protein. And you can see how it contains mostly beta sheets, and those beta sheets are held together by the backbone hydrogen bonding between adjacent beta strands. And so this provides the stability of the beta sheets, and it also allows the side chains to stick above and below to make a very conformationally stable conformation. So the next protein we're going to talk about is insulin. So insulin is a peptide hormone that's secreted by the beta cells in the pancreas in response to high blood glucose. So it's the main anabolic hormone in the body. So what it does is it'll bind to receptors on your cells and signal for the uptake of glucose by those cells. So it'll therefore decrease the blood glucose level. It'll also signal for the use of that glucose to basically produce glycogen, which is the storage form of glucose, as well as fatty acids and proteins. So it's a very small peptide, basically. So it's a peptide hormone. So it's very small. It's made up of 2 subunits, but together it only consists of 55 amino acids total. So it's only 6 kilodaltons big. And so here is the deposition of the insulin hormone that we're going to be looking at right now. So its code is 2HIU. It was actually, it's Homo sapiens, so it's human insulin gene, and it was actually made from that. And then it was deposited way back in 1996 by this group here. And instead of crystallography, this structure was actually determined using protein NMR. And there was 10 conformers submitted. So NMR gives you a more dynamic look at a protein structure because it can actually look at the movement of the structure in solution. So here's the structure of insulin, and as you can see, it's quite small compared to the proteins that we were just looking at, but it is made up of a combination of alpha helices and these disordered loop regions. So we're currently looking at the cartoon structure of the protein, which is a really easily interpretable structure. It shows you the alpha helices and the beta sheets, and as well as turns and loops. But you can also look at other types of structures using the program that I'm using as well. You can visualize the protein just by its backbone with the lines as shown here, or you can just look at all of the individual atoms that make up the protein using the stick function here. So this shows you the actual positions of the backbone and the side chain atoms, although hydrogens are generally left off for clarity. You can also look at a space filling model. So this sort of shows you the van der Waals radius of each of those atoms and the space that they're filling up in three-dimensional space. But the normal way to look at these proteins is using the cartoon model. That's the most familiar to most scientists and investigators. So as I said before, insulin is actually a peptide made up of two different chains. So these are two different peptides that are joined together to form this structure. So one chain shown here is in pink and the other one is in turquoise. So you can see in this one chain, it forms a small alpha helix at the N terminus, followed by a loop and another small alpha helix, and then terminates at the end. So it's a very short peptide. And the other chain has a loop region, which is connected to a sort of medium or small size alpha helix, and then ends in this extended sort of loop region at the C terminus of that peptide. So if we now show the side chains, which are the carbons are colored the same way as the protein, but you can see that the side chains are sticking out. Of course, the carbons are colored the same way as the peptide that they're part of. The nitrogens are blue, the oxygens are red, and any sulfur atoms are yellow. And this allows you to look at the interaction between how these two peptide chains actually interact with each other. So the most important way that these two chains are held together in the insulin protein is through disulfide bonds. So there's 3 disulfide bonds in the insulin structure. So there's one shown right here that is a disulfide bond within one of the chains. So you can see both of the connections are turquoise. And then there's another disulfide bond, and this is actually between the chains. You can see chain A in turquoise is connected to chain B in pink by this covalent disulfide bond. And there's an additional disulfide bond between chain A and B shown here. Another feature that's important for the sort of joining together these two chains is that the hydrophobic residues, which are shown here in gray, are clustered on the interior of the protein. And this makes perfect sense because this is a soluble protein. It's going to be transported through your bloodstream. So the hydrophobic residues are going to be clustered at the interior so they don't interact with water. This also helps bring stability between the interactions between these two chains. And so that's just an overall view of how the insulin molecule, how its structure, how the interaction between these two chains makes up the entire insulin hormone, peptide hormone. But of course, this peptide hormone actually does bind to the insulin receptor on the cells. That's what ultimately causes it to cause an uptake of glucose into cells and the synthesis of glycogen, fatty acids, and proteins. And so here is the actual structure of the extracellular portion of the insulin receptor bound to several insulin molecules. And so that's the insulin protein, and it is a small hormone peptide, very small, but very, very important. And we can learn some things structurally about it, including how the disulfide bonds hold and the hydrophobic residues hold those two chains together. So the next protein we're going to look at is myoglobin. So myoglobin is a single subunit oxygen binding protein related to hemoglobin. So myoglobin is very similar in structure to the subunits of hemoglobin, but it doesn't have any quaternary structure. It only has a single subunit. So it actually binds oxygen stronger than hemoglobin, and it does that by using an iron atom bound to a heme ring, which is a type of porphyrin ring. And this heme ring is used as a cofactor, also called a prosthetic group, because it's always bound to the protein, and it allows this specific protein to bind oxygen. And it was actually the first protein structure ever determined. So it was determined in the 1950s by crystallization and X-ray diffraction and first reported in 1958. And so here's the PDB deposition of the actual protein that we're going to be looking at today. So its code is 1MBD, and it's the X-ray structure of sperm whale deoxymyoglobin. So this is a myoglobin from a sperm whale, and it's deoxymyoglobin because it's not actually bound to oxygen. Sperm whale is actually my favorite animal, and so whales and other diving mammals actually have a lot of myoglobin in their bodies to help them bind oxygen and do those deep dives. So this structure was actually deposited all the way back in 1981 by this group, and it was determined by X-ray diffraction, and its resolution is 1.4 angstroms, which is very, very high resolution. So here's the structure of myoglobin here. You can see it's a single subunit, and it's made up almost entirely of alpha helices. So there's no beta sheets whatsoever, all alpha helices connected by loop regions. And you can see this heme molecule here, which is the cofactor or prosthetic group for the molecule. It's what allows it to bind oxygen. And it's shown here in green with the iron atom shown in the middle as a red sphere. So here's a little bit of a close-up of that heme molecule. You can see it makes a cyclic ring, and then there's 4 nitrogen atoms shown in blue that actually coordinate that iron molecule in the center of the heme molecule. And that's actually what's going to bind to the oxygen. And so that heme molecule is actually always present in the myoglobin structure. It doesn't go away, but it can either be bound to oxygen or not. So how is that heme molecule integrated into the active site of myoglobin? Well, there's several protein interactions that are involved with that. You can see this serine molecule here makes bonding with the heme molecule, as well as this histidine molecule here and this arginine molecule here. So there's some other features I'd like to point out about myoglobin. One is that there are several hydrophobic patches that are, of course, on the inside of the protein. So you can see one of them here in gray. And then here's another hydrophobic patch in gray at a separate part of the protein. So both of these hydrophobic patches are clustered on the interior of myoglobin. So with myoglobin, it's a good example of looking at tertiary structure. So we talked about how secondary structure is maintained by hydrogen bonding within the amino acid backbone, whereas the tertiary structure is going to be dictated by the side chains. So we already saw how the hydrophobic side chains are going to be clustered at the interior. That's a big part of tertiary structure. There's also residues that are going to interact within the protein that are going to help stabilize and make those secondary structures interact with each other. For instance, here is a salt bridge between a glutamate residue and a lysine residue. Here's an interaction between an aspartate and a histidine residue. So these are holding these two different alpha helices together in 3 dimensions. And then here's a third one with another aspartate residue that's interacting with an arginine. So these are located on separate alpha helices and help to hold this protein and give it its overall tertiary structure. And so that's myoglobin. It's a very famous it's the first protein ever determined its structure, and it's made out of entirely alpha helices with this cofactor heme group, and you can see how the side chains are involved in maintaining the tertiary structure, its overall 3-dimensional folding in space. So the next protein we're going to look at is collagen. So this is a structural protein that provides support to different connective tissues in your body, so your skin, your bones, cartilage, tendons, and ligaments. It's actually the most abundant protein in mammals. So more proteins in your body are collagen than any other proteins. And it forms these long fibrils, which you can kind of see here, which are made of triple helices of repeating units. And so here's the deposition of this protein. So its code is 1CAG. It's a collagen-like peptide, so there actually is a few modifications, but this is basically the structure of collagen. It was deposited way back in 1994. So I just got these codes out of your book, and they use a lot of old structures in there, apparently. That's okay, though. They're still very good. So it was X-ray diffraction determined by X-ray diffraction to 1.85 angstroms, which is very, very high resolution. So here's a picture of the structure of collagen, and you can see how it forms these triple helices. So each of these polypeptide chains in blue, pink, and yellow are individual polypeptide chains that will interact with each other, wrap around each other to form this triple helix. And so this can make very long fibrils, longer than the peptides that we see right here. They can go on and on for a great length. Now we can actually also just look at the individual residues and amino acids that make up these polypeptide chains, as you can see right here. Now if we focus in on the individual chains, we can see that there's a repeating unit for all of these polypeptides. So right here is actually a modified amino acid. It's a hydroxyproline. So you see this oxygen atom right on top. That is added onto proline to make it a hydroxyproline. And that modification comes from vitamin C, which is why you have a vitamin C deficiency. You have problems with your connective tissues in your body, because that hydroxy group on that modified proline is actually going to interact with connective tissues to provide even more support. So we see here that we have a hydroxyproline, followed by a glycine, followed by a regular proline. And that's followed by a hydroxyproline, then another glycine, then a proline. And these glycine residues are actually really important for maintaining that triple helix. So we can see here 3 glycine residues highlighted in yellow, all on different polypeptides in this triple helix. And you can see that they make actually hydrogen bonding interactions with each other. And this is what gives that triple helix such structural support and allows it to do that function in your body. So that's collagen. It's the most abundant protein in your body, and it's made of these long fibrils consisting of these triple helices of repeating units. So the next protein we're going to look at is called CD4. So this is actually a glycoprotein, which means it's a protein that has carbohydrates on it. And it's an immune protein that's a co-receptor for T cells, which are, of course, part of your memory immune system. So there's T cells and B cells. So those make up your adaptive immune system. So it assists T cells with communication with antigen-presenting cells. So those are cells that present foreign material that you need to generate antibodies against. And the point of us looking at it is to look at protein domains. So it contains 4 different immunoglobulin domains, or Ig domains. And if you'll remember back to our lecture, these are regions within the protein that fold independently of the entire protein and may have independent functions. And so here is the PDB deposition of the structure we're going to look at. So its code is IW1WIO. So it's a T cell surface glycoprotein, CD4. And it was it's from Homo sapiens, so this is a human CD4 protein. It was deposited again a long time ago in 1996 by this group. It was determined by X-ray diffraction, but the resolution's a bit lower, so it's 3.9 angstroms. So that you can get some of the general backbone of the features, but you may be missing some side chains in the structure. And so here we have the protein CD4. So it's just shown in gray right now, but I really want to emphasize that there's different domains in this protein. So there's one domain, so domain 1 here in red, domain 2 here in yellow, domain 3 in blue, and then domain 4 in orange. So each of these individual domains will actually fold independently or semi-independently from the other domains and thus have possibly a different function. So that's CD4, and we just want to use that to emphasize the importance of domains in different proteins. So a lot of different proteins have more than one domain. They can fold independently, and they can often have different functions in the cell. So the last protein we're going to look at today is hemoglobin. So hemoglobin, which you've probably heard of, is an oxygen carrier protein that's present in high concentrations in erythrocytes or red blood cells. So it carries oxygen to the cells, which is essential for aerobic respiration. It also carries some of the CO2, which is a byproduct of aerobic respiration, back to the lungs to be exhaled. So hemoglobin has a quaternary structure. It's a heterotetramer. So there's 4 subunits in hemoglobin, and they function allosterically, which you can see from this GIF on the right. So oxygen binding at one subunit actually causes a conformational change that increases binding at other subunits. So it's a very well studied, very interesting, and very important protein. And so the structure we're going to look at today is PDB code 1A3N. So this is deoxyhuman hemoglobin. So it's human hemoglobin without oxygen bound to it. And it was deposited way back in 1998 by this group here. It was determined by X-ray diffraction, and it has a very good resolution of 1.8 angstroms. And so here's the structure of hemoglobin, as you can see here. Now, this is actually made up of 4 different subunits. There's 2 alpha subunits, which are shown here in blue, and then there's 2 beta subunits, which are shown in pink. So the interaction of these different subunits is what actually makes the full functional protein of hemoglobin. So it does not function the same way if all the different subunits are not associated. So what's holding these different subunits together? Well, there's a lot of different factors in that, but we can look at some of them right now. So in this region right here, you can see that a aspartate residue right here is interacting with a tryptophan, and this arginine residue is interacting with this glutamate residue in this region here. So the tryptophan and the glutamate are on one subunit, and the arginine and aspartate are on another subunit. Then we have this other position where a lysine residue on one subunit is interacting with the carboxy terminus carboxyl group of this histidine residue on another subunit. And then lastly, we have this histidine residue on one subunit interacting with a serine residue on another subunit. So these types of interactions are what make the different subunits of hemoglobin interact so strongly to make this fully functional protein.